Hello, my name is Alex Lindsay, and I'm from Ohio State University, and I'm excited to talk to you a little bit about the projects we've been working on to improve pennycress establishment for agronomic production throughout the U.S. Midwest. Some of the work that I've been leading with regards to the I Prefer project is in the area of seed treatment. And so I wanted to talk a little bit about the work that we've done as part of the I Prefer project, starting way back with our first presentation from Corey Lee and the first experience she had doing some exploratory research, all the way through our final projects that have happened at the end of our experiments. And so today I'd like to talk about the case studies with regards to using seed treatment and enhancements in pennycress to improve this crop's productivity in the U.S. Midwest. Seed dormancy is a natural trait found in flowering plants. It's very biologically important, and it ensures temporal distribution of germination in wild species in natural ecosystems. There's two main dormancy mechanisms that we can find in plants. They can either be external or exogenously applied, or endogenous or internally controlling germination of different seeds. Examples of exogenous dormancy or external dormancy are physical in nature, where something like the seed coat itself would restrict water uptake for the seeds. It could restrict oxygen diffusion, and it's typically overcome by breaking the seed coat in some way, either scarifying it with sandpaper or cutting it open, or dissolving it using something like acid, a strong acid. Chemical exogenous dormancy could be something like the seed coat containing compounds that prevent germination that need to be leached out before germination can occur. And you can overcome that type of dormancy using soaking with other materials like bleach or hydrogen peroxide. The more common type of dormancy found in most plants is endogenous physiological dormancy, where seeds have a balance of abscisic acid and gibberellic acid contained within their plant parts. Abscisic acid response predominates in dormant seeds by inhibiting germination, and it can be overcome by either stratifying seeds or holding those seeds in cool, moist conditions, or through after ripening in some cases where you have dry storage where the embryo matures. This is typically overcome by treatment with gibberellic acid hormone, the one that promotes germination, and we're just adding more of it at any given point in time. Endogenous morphological dormancy could be also overcome through stratification or after ripening, and it's typically because the embryo in the seed is not fully mature. And so that embryo actually needs to finish its maturation phase after harvest before that seed will be ready to germinate. Initial dormancy in some crops is good because it prevents germination on the plant prior to harvest, but you don't want it to last for very long. So having short duration, non-deep dormancy is good to facilitate harvest, but we want it to go away relatively quickly, or else it can cause issues with germination and establishment and when planting that crop the next year. Pennycress is still a new crop. Again, domestication efforts for this began in the 2010s, and innate dormancy is still an issue in some of the lines that we've been testing in research. And so my goal with this project was to look for ways to alleviate dormancy in early lines as we're continuing to improve genetically this crop moving forward. Germination is key. Without having solid stands from a crop production standpoint, we don't get a good yield. And potentially, if we're planting a bunch of dormant seed into the soil, we're creating weed seed bank issues in future years, and it could be a problem. There's multiple seed traits about this. It's a small, rough coat. And so researchers have been looking for ways to alter the seed shape and size and potentially look to improve methods for improving establishment. If this is a really small seed, could we increase the weight of it, improve uniform distribution by something like pelleting to improve establishment? And also, if it's susceptible to diseases, could treatment with a fungicide potentially improve germination and establishment? We don't really know necessarily as well what dormancy mechanism is overriding in pennycress because there's a lot of diversity in dormancy mechanisms found in Brassicaceae species. So we underwent the process of trying to better understand this seed by leveraging seed treatments and technology to improve establishment in this crop. When we talk about seed treatments, that's typically referring to chemicals applied to the seed for control of insect pests or pathogens. In this case, our main focus would be on fungicide application because damping off and fungal infection of seedlings was more of a problem at this point in time than feeding from insects, where an insecticide would be beneficial. And we also weren't ready to get into some of the biological controls that are on the market because we still are just trying to get a handle on this crop. Seed enhancements, on the other hand, are techniques applied to seeds to enhance germination after harvesting and conditioning. Some of the examples of this would be using film coating applications, biological inoculants, in the case that we see for things like legumes, but also priming seeds to increase germinability at a faster rate, or applying things like a seed pelleter using a rotary coater is kind of what we can see going in this sample here. We have pennycress seeds rotating in the drum, and when the lid is closed, we can actually add liquid and solid materials to build those seeds up from a size perspective and increase the weight of an individual seed unit. And so throughout this talk, I'm going to go through 5 different phases that we've undertaken in the investigation of seed treatment in pennycress. And so I'm going to begin talking about the initial assessments we did in 2020. Corey Lee was an integrated plant systems undergraduate researcher through the iPrefer program that worked within my lab to kind of look at some of these initial questions. We received a black seeded pennycress type for a lot of our agronomic work. It was line MN106. An untreated seed germination rate was 71% under good conditions. This means that of the seed we were receiving, about 30% of it was not germinating. And so we wondered, could we increase germinability of this species by seed treatment? So we ended up employing 14 different treatments, 3 of them being scarification, either with sandpaper, treatment with hydrogen peroxide solution, and also treatment with gibberellic acid solution. And for our hydrogen peroxide and gibberellic acid treatments, we ended up soaking them for different time frames. We didn't know if a shorter soak would be better or a longer soak would be more beneficial. Once the seeds were treated, we either placed them directly into the germination plates, or we allowed them to dry for about a week before plating them because we didn't know if that initial hydration phase would increase germination or not. Some of the initial results we saw with this black seeded line was that scarification increased germination from 71% to 79%, and that was a statistical increase that was significant. But treatment with hydrogen peroxide negatively affected germination. Any duration, 6, 12, 18, 24 hours of soaking in those solutions decreased germination, which is not beneficial from a productivity standpoint. Treatment with gibberellic acid, regardless of the timing of that soak, did improve germination. And so some of the conclusions here was that hydrogen peroxide was probably not a treatment we would recommend to increase germination. And both scarification and GA treatment did increase germination rates, but we need to vary the concentration of the solutions that we used, as well as the time that those plants are exposed to that solution. We also learned that drying the seeds after treatment did not reduce germination, which means these could be viable options for use in industry as a seed enhancement practice. So that kind of moved into our next version, was to see if seed treatment or enhancements improved germination in the wild type pennycress line. So in this case, instead of just focusing on GA solution and peroxide, we ended up adding in a few different enhancements. We decided to utilize a seed treatment of fungicide treatment of fludioxonil. we again had our hormone treatment of gibberellic acid soaking, but then we added another enhancement called pelleting, where we either used pelleting by itself, pelleting with a fungicide, or pelleting where we added gibberellic acid into the binding solution used to build our pellets. The idea there was, could you treat the seeds with a little bit of hormone, but use less than you would if you were using a full-on soaking treatment? So the fungicide we used was fludioxonil at a rate of 50 micrograms AI per gram of seeds, which is the use rate recommended for canola. And we applied it again using our rotary coater, but instead of building a pellet using the drum, we actually applied the fungicide through the tube and used the atomizer to evenly distribute it on those seeds. For the hormone treatment, we again used gibberellic acid. We used a lower concentration of gibberellic acid solution in this experiment, and we ended up soaking those seeds for 12 hours. We rinsed those seeds off after removal and then allowed them to dry before starting our treatments. To build our seed pellets, we ended up using a filler of diatomaceous earth in rotation with a binder solution of this Seedworks Stix Light solution. What this does is we add the binder solution first, allow the seeds to get coated with this wet solution, add in some light powder dusting, and then repeat those steps over and over. Periodically, we'd remove the seeds and sieve them to remove extra or excess material that didn't stick to the seeds. And we continued to increase the size of the seeds until we went from 40 gram starting sample to a final value of 130 grams in that material. After we were done, the seeds ended up looking like what we see here on the screen. We repeated that process instead of just pelleting them alone with the binder and the filler materials. We ended up adding gibberellic acid in one treatment to the binder solution to see if adding that liquid in small amounts, but having a little bit of GA would improve germination. We also looked at adding a fungicide during the first pelleting cycle to get the fungicide in contact with the seed, and then we built the pellets around that. And we also looked at improving stability of the pellets using carnauba wax at the very end of the pelleting process. So we added a little bit of this wax emulsion to the last two cycles to see if it would improve pellet integrity. Once all of our treatments were applied, whether they were soaked or pelleted, we allowed them to dry in an airflow chamber at 30°C for 15 hours, and then removed them from the chamber and allowed them to dry at room temperature for 3 days. Until they were tested for germination, we kept them in cold storage in paper envelopes. So with this experiment, again, we had all of our treatments applied. We had two different GA soak treatments, a 0.01% or solution and a 0.05% solution. And we had all of our different pellet treatments applied as well. To assess changes in seed weight after treatment, we weighed 1,000 seeds from each treatment and also used sizing to see how we increased seed diameters using nested daughter sieves to see if pelleting increased our seed size uniformly. The last thing we did was we agitated seeds on a mechanical shaker for 10 minutes to see if physical agitation negatively affected pellet integrity. And what we observed was that the fungicide treatments, GA soaked solutions didn't change seed size and seed weight, which was expected. And we did see a significant increase for all of our pelleting treatments at a similar weight and size diameter gain. So our uniformity of pellet development was very similar regardless of the pellet process. All pellets held up very well to the shaking test, meaning that they all exhibited similar integrity from that agitation assessment. So the weight increased by about 140% when pelleted, and size increased by about 30% from a diameter perspective for each of our seeds. Interestingly enough, the best increases in germination occurred when we treated our wild type seeds with gibberellic acid, whether that was as a GA soak solution or if gibberellic acid was added to our pellets themselves. That was when we had the best final germination. Pelleting our seeds with and without wax resulted in moderate gains to germination, but just treatment with fungicide alone was similar to the non-treated control. In this case, our germination of our untreated was only 30%, whereas treatment following gibberellic acid exposure exceeded 90%. So the conclusions from this is that seed pellets were uniform. We were pretty good at making these pellets in the lab. Seed treatments did improve the final germination in some cases. GA treatments were most effective. Seed pelleting did improve germination, but was not quite as effective as the GA treatment. And so under ideal conditions where these were performed, germination characteristics would be the same, but increased germination with seed treatments can be beneficial for better establishment in field conditions. And so that kind of led us to our next experiment where we wanted to look at, okay, we know that we can increase germination in the lab, but what about in the field? Would field establishment of pennycress improve with our seed treatments? And so we collaborated with the different I-Prefer institutions and ended up establishing pennycress sites at 3 locations in Ohio, 2 in Illinois, 2 in Wisconsin, and 2 in Minnesota. And we ended up going back to the golden seeded, which is kind of the newer commercial version, and our standard black seeded that we had been using in the previous experiments. We narrowed our treatment list down to a few treatments. We had an untreated control, a GA soak treatment at 0.01% GA solution, fungicide treatment only, and then 3 pelleting combinations, 1 with fungicide, 1 with fungicide and gibberellic acid, and 1 just pelleting using the procedures that we discussed previously. We had a uniform seeding rate in each plot, which corresponded to roughly 5 pounds of live seed per acre. And we ended up hand broadcasting these. So the seeding rate might have been a little bit lower than we would recommend for commercial production if you're broadcast seeding. But again, this was small plot research with limited seed supply. At each location, we recorded our planting date, previous crop, and surface crop residue to see if that had any impact on establishment. Throughout the season, we ended up collecting autumn establishment values using percentage green area cover using the Canapeo app for a smartphone device. What that does is it takes a picture of a known area. We'll be able to identify what parts of the picture are green and convert it into a percent area value. We quantified spring survival of our pennycress in the same way using Canapeo. And then at harvest time, we measured stem lodging, seed yield, and 1,000 seed weight from each one of our plots. In general, what we saw was that as we measured autumn establishment, GA treatment really did improve black seeded establishment in most cases. If we look at the Min 106 NS, we saw a significantly greater percent canopy cover and stand counts when treated with gibberellic acid here, as well as when gibberellic acid was included in the pellet itself. Pelleting also did increase fall establishment for the black seeded type. The untreated type of our golden seeded ended up having the greatest establishment of the treatments here. Fungicide was slightly greater from a percent area perspective, but pelleting really did not improve establishment. In fact, pelleting negatively hurt stands for our golden seeded values. What was interesting though, across all of our locations, fall establishment was fairly slow, and percent canopy cover at this point in time was 5% or lower in all treatments. We saw very similar trends when we established a spring survival measurement, though spring canopy coverage did increase beyond what we previously had recorded. The best canopy coverage was seen between 14 and 17% for our black seeded lines. This occurred when we treated with gibberellic acid and pelleting, but the golden seeded established best with no treatment alone or fungicide by itself. Again, pelleting was seen to negatively affect establishment in the golden seeded line. We saw no difference in seed treatment or genetics really when related to lodging percentages. There was no interaction with our varieties tested. And we did see those trends for autumn survival, spring survival, and translate into yield differences. Golden seeded lines yielded highest with fungicide treatment or non-treated lines in general here. GA treatment improved seed yield in our black seeded lines the best. Survival and establishment for our yellow seeded was negatively affected by pelleting, and we saw that come through in yield. We saw a 25 to 50% yield reduction in our yellow seeded lines when pelleted as compared to when untreated. So in general, GA treatment and pelleting did improve black seeded establishment and germination in the field. GA treatment did not improve establishment of the golden seeded type, but it did exhibit similar yield to untreateds, and it's not likely necessarily needed to improve establishment and yield in field environments. Pelleting of golden seeded types reduced establishment and yield, and it's not an advisable practice. And fungicide may help to improve establishment of the golden seed type. And we may want to look more into this in the future and do more investigations as far as how that fungicide may be improving establishment before fully recommending a prophylactic fungicide seed treatment moving forward. The next question was looking back at this idea of storage over time, and could seed treatments be applied well in advance of planting, and how might this impact germination and survival? To look into these treatments, we had 4 different types. 3 of them were black seeded types. 1 of them was a golden seeded type, which is referred to as CB2. We had GA soak solution, a pelleting treatment, and then pelleting plus a reduced percentage of GA solution. And we ended up storing these seeds for 0, 1, 3, 9, or 12 months to look at that long-range survivability. We stored them under cold conditions and room temperature conditions, but the results are fairly similar regardless of the temperature. So I'm only going to present results from one of our temperatures. And basically what we saw was that after 3 months of storage, most germination percentages were fairly similar to what they were when seeds were initially treated. So that's good news. But over time, germination values tended to decrease for most of our lines, especially those that had been pelleted. So if pelleting is a practice that's desirable for a black seeded type, those pellets really should be only stored for roughly 3 months at a time before viability really starts to be negatively affected in those seed lines. Germination, when treated with gibberellic acid, was much improved for our 3 black seeded lines compared to the blue untreated lines, regardless of the cultivar. So the black coat pennycress may still have some dormancy controls in place, and these results suggest it may be physiological in nature and could be overcome with a very small amount of gibberellic acid exposure. The golden seeded type, though, was impeded with gibberellic acid and pelleting, as well as pelleting alone. And again, it's a non-advisable practice. Pelleting had more evident decline after 9 months than other treatments, particularly in our golden seed type. In general, gibberellic acid soaking improved the germination speed index or seedling vigor for all varieties. That's kind of what we saw here. So it's possible treatment of GA didn't improve overall germination of our golden seed line, but it may have sped up germination of that, allowing for more rapid emergence, which may be beneficial in field type environments. GSI was inhibited by pelleting CV2. That's kind of what we saw in panel 2 here. Cultivar 2 was impeded because of that pelleting process. And over time, vigor indices or the speed index declined for all treatments across all varieties. And so really, if we're going to apply seed treatments, they really need to be done and planted within 3 months of application. Longer storage decreased viability and vigor over time. Gibberellic acid improved seed vigor most in black-seeded pennycrests, while it was ineffective in the golden-seeded lines, though golden-seeded lines treated with GA did exhibit a high germination speed index. Seed pelleting without storage under low temperatures or under good temperatures increased germination in black-seeded lines, while it reduced germination and vigor in golden seed, but storing for longer than 3 months is not a recommended practice. And that leads us to our final focus that's been most recently published in Seed Science and Technology. And we question this idea. We've been building these pellets with multiple components. We've been working on these treatments and very complex values, but could we get similar results if we simplified the process? What step in this pelleting process is really giving us the biggest bang for the buck from a germination perspective? For seed treatment and pelleting, we're really looking at 2 different components. Physical agitation of the seeds could be improving germination by providing some sort of scarification. Again, going all the way back to 2020, scarification did improve germination marginally in some of our pennycrest lines. So could this be contributing to our overall gains in germination? The other idea is that when we treat with fungicide, we're applying it with a small volume of liquid. And could that small volume of liquid application provide some sort of priming effect and be contributing to increasing germination? And could we gain similar gains in germination if we just added the volume of liquid instead of adding it with a fungicide? And so we looked at the seed treatment steps, looking at untreated seeds, comparing that to running it through the rotary coater physically, running it through the physical rotary coater with distilled water or the liquid itself, running it through the coater with the pelleting binder solution only, which was applied at the same volume, or using the scarification process with the binder with the fungicide, which is how our fungicide treatment has been applied through all of the past research that's been discussed. And what we observed was that all treatment steps slightly increased germination of black-seeded lines, though all of these were non-significant at both the 7 and the 10-day phase. So regardless of what was being done, slight agitation of the seeds or slight agitation with some water marginally increased germination, but it wasn't significant. We found that for the golden seed, the only thing that really started to impede germination at 7 days or slow down that germination speed index was the application of the binder and the binder plus the fungicide. For some reason, that seemed to start to slow the germination process during the 7 days, but it had no effect on final germination at 10 days. It's possible that having extra layers of material was just more physical restriction for that seed to germinate through and emerge through, which could have been slowing down that initial germination process. So a similar experiment was conducted to look at the steps of seed pelleting. And was it something early in the process where we can terminate pellet formation before we reach that 120 gram threshold that we had been targeting, going from 40 grams to 120 grams? Could we lessen that time in the pelleting process and still gain the benefits that we saw from a germination increase from the pelleting material? So we started by running the scarification process 3 times, running the first 3 steps in pelleting, doing that with water addition, and then doing it with the binder solution. So kind of doing the initial steps to see if it was a slow priming effect, increasing germination, or if full-on pelleting was needed to improve germination. And what we saw was that the scarification with water addition did slightly increase germination for our black seeded line. We saw more benefit from the binder solution, suggesting there could be some interaction there of a hydropriming effect early in that process. So it's possible we may not need to build a full-on pellet unless that's desirable from a seeding and seed handling perspective to increase germination. For our golden seeded lines, we really saw no influence of any of our treatments from the initial process affecting germination. The only thing that did affect germination was pelleting. We saw 0% germination in this seed lot after being pelleted, which further kind of hammers home that idea, don't pellet golden seeded pennycress at this point in time. The final step would be thinking about GA treatment and thinking about optimizing that hormone treatment. In past work, we've looked at anywhere from 6 hours soaking through 24 hours soaking. We backed down the concentration and had been using a time of 12 hours. But we wondered, could we get away with a very short duration of 30 minutes, 1 hour, or 4 hours? The other idea would be looking at other hormone choices. To date, we had been only really looking at gibberellic acid, but there could be some other hormones that could potentially improve germination as well. So we again had untreated seeds. We soaked these seeds in distilled water for 30, 60, or 240 minutes, soaking in either 60 or 0.01% GA solution, or we found another hormone that had been shown in literature to potentially improve early season germination and growth called benzoadenine or BA solution. So we tried that as an alternate treatment. We looked at both black seeded lines as well as 2 separate lots of golden seeded pennycress as well. There's a lot going on this chart, but I'm going to work to synthesize this for you right away. Looking at these here, the blue bars are our black seeded lines. The orange bars are the new harvest of the golden seeded line, and the gray bars are the old harvest of our golden seeded lines. What we observed was that soaking solely in water hurt black seeded germination. We saw declines in germination of black seeds when soaked in just water. But when soaking in gibberellic acid solution for at least 30 minutes, resulted in improved germination over our control. When we soaked seeds in BA solution, which would be the last bars here, we really didn't see any improvements in germination. On the whole, TT8 or the golden seeded lines, regardless of harvest year, really were not responsive to our treatments. And this is very similar to the work that we've already reported on and kind of further emphasizes that it's unlikely that seed treatments will improve overall germination in those lines. So with that, we'd really like to thank our funding from the USDA NIFA program for the Sustainable Agricultural Systems Grant, and thank our investigation partners. Again, throughout this presentation, I've referred to multiple publications that we've generated in results from this research. And I hope you've enjoyed listening to this presentation. You've learned a little bit about pennycress emergence and seed treatment, when it might be needed, when we might be able to get away with using non-treated seeds. And again, thanks for your time. If you're interested, these slide sets are available on the same site where you accessed this link. And we've developed a seed dormancy lab that could potentially be used in K-12 classrooms or undergraduate classrooms to further investigate dormancy mechanisms and how we can relieve those using applied seed enhancements. Thanks again for your time, your attention, your interest, and best of luck learning about pennycress. If you'd like to reach me, I can be reached at lindsay.227@osu.edu. Thanks again for your time. Goodbye.